Good afternoon, everyone. Thank you. And I'd also like to thank Spiros for inviting me today. And this came out of a meeting where we were both at in California for something called Diva Day, which was a workshop around a piece of software called Diva, which is produced by Solema. And I'm going to come to that at the end. We've been working with them on a new advance in light modeling. But Spiros asked me to talk about some of the science behind the work we've been doing on light applications. And so I'm going to be talking a little bit about using light in a way that you probably haven't used it before. It's not about seeing, it's about what light does to the brain. And so I'm going to talk about some of the background neuroscience of why we're interested in this, how the system works, and then talk a little bit about application, and then a little bit about the new modeling tool we've developed. I have to show you my conflicts of interest disclosure. I'm required to. It's a big, long list. It looks like it should be a lot of money, but it isn't. Yet, but I do consult for a number of lighting companies around light and sleep and jet lag, but we want to make sure that the application of the basic work we've done is correct, and that's why I get involved with industry. And so why do we care about light? Why do neuroscientists and sleep researchers care about light? Well, it's because we've evolved in a very strong light-dark cycle. Our biology is driven to a great extent, or at least affected to a great extent, by light and dark. And so if we want to understand how to be as healthy as we can be, as productive as we can be, light is a really key component to that. And you can think of light as having a whole range of what have been termed non-visual effects. I know it's not very good to call something by what it isn't, but we're trying to make people think about a separate system from vision. This is not about seeing. This is about light information getting to the brain. And so there are multiple hormonal, there are multiple biological, multiple effects of light on our physiology that are separate from vision. And so here are just some of them that are listed. Does that? Well, it doesn't matter. So you can see in the top, we have many of these direct acute effects of light. Light is a stimulant. Light alerts the brain directly. You can think of it a little bit like caffeine in that regard. And so we can use light to make you feel more alert. We can use light to change your brain activity so it's more alert. We can improve your performance using light. But we also can see changes in other systems that are helping to tell the brain about night and day. So there's a hormone called melatonin, which tells the brain it's nighttime. You only produce melatonin at night. But if you see light at night, the brain thinks it's daytime and suppresses that melatonin. Light can also induce cortisol at certain times of day, the fight and flight hormone, which gets the body ready for working, get it ready in the daytime. If you see light at night, that goes up because the body thinks it's day. Your heart rate goes up, your temperature goes up. There are many effects of light separate and apart from what we see. And then we can think about light having a circadian effect, an effect on the biological clock, our 24-hour rhythm. And so we need to keep our clock synchronized every day by the light-dark cycle. And when we shift our light-dark cycle, if we're a shift worker or if we go across different time zones or even seasonal changes in light-dark exposure, then we need to reset our clock and change its timing. And light is the time cue that does that. And then there are others. If you're an animal that breeds seasonally, like sheep or ferrets or goats, this is driven by a light-dark cycle. Butterflies that navigate to Mexico are using this same clock and light. Much navigation by birds is using the same system. So there are many, many things that light does other than help us see. And certainly in humans and mammalian species, light is the most powerful time cue for resetting that clock. And what the clock needs is a regular light-dark cycle. It's expecting the natural light-dark cycle, which doesn't change much day to day, a few minutes from day to day. So the clock wants stability, the same light-dark cycle every day. And if we do that, if we're exposed to that, our clock synchronizes itself and everything that the clock controls is realigned. But if we don't do that well, if we vary our sleep-wake and therefore light-dark cycle every day, if we travel across time zones, if we go to bed 2 hours later on a Friday and Saturday night and then try to get up on a Monday morning, we can't because that light has shifted the clock and put us out of sync. It's desynchronized us. So if we get it wrong, if we get the light-dark cycle wrong, everything that the clock controls is wrong. And that means sleep, fatigue, performance, mood, metabolism, there are real issues. And shift workers, if you think of shift workers as the most extreme example of a shifted clock day to day, shift workers do have more diabetes, more heart disease, more cancer, more depression, because this continual shifting of the clock causes problems. Now, you and I may not be shift workers, we may not have that extreme change, but even that wobble day to day is having an impact. And so the environmental light, the light you expose yourself to after dusk, that electric light is having a major impact. But if we think about how we might use light, and for most design applications, it's the direct alerting effects of light that are going to be applicable. You're generally not trying to reset rhythms or treat disorders in a built environment. We're trying to make people perform as well as we can. And so think of light as a non-pharmacological sleepiness countermeasure. It's safe, it's short-acting, it's reversible. We know we need it because we're all using massive amounts of caffeine, and so we need a stimulant in our lives, sadly. But anywhere where there's a light bulb is a place you can do a better job with light. Because I've listed a few here, you can think of many more, places where light will help people perform. So in offices and schools, we want people to be alert and learning well. When people are working in safety-sensitive occupations or in healthcare, we want the staff to be working well and most alert. So anywhere where we want to have people alert and functional is where we can do a better job. But the challenge, of course, is to bring this into design. It's no good me doing experiments in the lab unless it gets translated. And so we have to bring what we've learned about light into design to optimize both the visual and the non-visual benefits. Of course, you still have to see, and you still have to design a space well to see by. But we need to incorporate now these non-visual effects at the same time. So I'm going to talk mainly about wavelengths today, just for sake of time. But if we think about the properties of light that we have to consider when thinking about this non-visual system, there are a number of them which you would also be considering, of course, for visual responses. Light intensity, light timing, the pattern, your light history. But light wavelength, the spectrum of light, has really been the major breakthrough in our field in the past few decades in terms of a change in our approach. And so, to reiterate, you can think about the eye as having two functions, just like the ear. The ear helps you listen, the ear helps you balance, and they're separate systems. The eye is the same. You have an eye to help you see, but you also have an eye to tell the brain what time of day it is. And as you're going to see in a moment, that's a different system in the eye. And so we can think about the eye doing two things, telling us, allowing us to see the environment, but allowing us to know time. And these non-visual effects are driven by a separate photoreceptor than the rods and cones you used to see. So there's a non-rod, non-cone photoreceptor in the ganglion cell layer of the eye, and I'm going to come to that in a minute. And it contains a pigment, a photopigment called melanopsin, and that melanopsin is most sensitive to blue light. And its peak sensitivity is at 480 nanometers. And so this is why you've heard about blue light, and you've heard about people using blue light and changing blue light content, changing the color temperature of light, although again we'll come to that, because people are trying to either increase or decrease the stimulation of this photoreceptor, which has a peak sensitivity of 480 nanometers. So if we take now a cross-section through the eye, light is coming from the top, passes through the ganglion cell layer, through the eye to the rods and cones at the back of the eye. Those rods and cones detect light, send signals back down through the ganglion cell layer, down through the optic nerve. And until fairly recently, the last couple of decades, we thought that really all of the effects of light were driven through the rods and cones' ability to detect light. But experiments were done in sort of natural experiments in blind humans and experimental models where the rods and cones were systematically knocked out or removed from various animal models. And yet people still got circadian and alerting responses to light. And nobody understood this. And in fact, when it was first discovered, everyone thought it was wrong. Because we've been studying the eye for thousands of years, everything goes through the rods and cones, how can there possibly be another photoreceptor? And so no one believed it, but the evidence built up and built up. And so you can have a system where there are no rods and no cones, and yet the circadian system will still respond to light. And in 1998, a researcher called Iggy Provencio in Virginia was looking for the photopigment that controls the movement of melanin in frog skin. So frogs can change color, they can move the melanin content, and he was looking for what the the light detector system was that controlled that. And he discovered this opsin called melanopsin. And then he looked for it in mammalian eyes. And he found it in a very small percentage of retinal ganglion cells. And this is remarkable, and still is a remarkable discovery, because it turns out that a very small percentage, less than 0.5% of those retinal ganglion cells are light sensors. And they've been called IPRGCs, intrinsically photosensitive retinal ganglion cells. But there's very few of them. But these are the blue light sensitive cells that are in a different part. The rods and cones are here, they're gone. It's these ganglion cells that detect light, and no one had ever discovered that before. And these cells are in fact hardwired straight to the clock in the brain. This is the underside of a rat brain, the eyes are here, and this is the site of the biological clock. We have this nucleus in the hypothalamus where the clock rhythms are generated. And about 80% of the projections from these cells go straight to the clock. This is what it's evolved to do. And if you look at the distribution of melanopsin across the retina, this is a primate retina, a flat mount of an eye to see where these cells are. They're across the entire retina. They're not focused centrally like we might have for vision. They're across the whole retina. And in fact, the dendritic fields of those cells are also contain melanopsin. And so what you can think of this system is it's a net. It's a network of light detection across the retina. You don't have to look at it. It doesn't matter if the light's indirect. As long as the light's getting into the eye, the brain is detecting that. And it's relatively insensitive because the brain doesn't really need to know much really about the intensity. It needs to know whether the light's off or the light's on. Is it day or night? Is it winter or summer? And in fact, this system is older than vision. Melanopsin works much more like an invertebrate opsin. It evolved before vision. This is a much more fundamental aspect of our biology than vision, which came later. So we've got this novel non-rod, non-cone photoreceptor that's most sensitive to blue light that is telling the brain about time, essentially. And if you do an action spectrum for the different systems to try and work out what's the sensitivity in terms of the spectrum to these cells, it doesn't matter if you look at a rat cell in a dish or a primate cell in a dish or a pupil response of a mouse or the pupil response of a human, all of these responses peak at 480 nanometers in the blue visible range. So it's very consistent across these, across mammals. And you can see here in the human data, if we plot at the same time the sensitivity of the blue, the short wavelength cone, the rods, the medium and the long wavelength cones, you can see that melanopsin, the IPRGCs, don't match any of those known photoreceptors. And so this is the photobiological evidence that this is a different photoreceptor to the rods and cones we used to see. And so we have this novel non-rod, non-cone blue light receptor in the eye that is working, that can work completely separately from the visual system. Now, I do study blind people with different types of blindness, and we have done a lot of work to understand that impact. But in most of our experiments, we can't knock out the rods or cones. I have to use different wavelengths of light to try and work out indirectly where these responses are being generated. And so the first work to show that back in 2001 from Thomas Jefferson in Philadelphia and the University of Surrey, where I trained, they published in the same week 2 action spectra for melatonin suppression, which is one of those acute responses to light. And you can see that both of them showed a peak in the blue range around 460 nanometers in their experiments, and we can talk about why it wasn't exactly 480. But the point is that it doesn't match either the rod, the scotopic, or the cone photopic function. So these were the first example that functionally these responses of light were not driven by the rod and cone system. And since then, there have been many, many studies. I'm just going to show a few of them showing the principles, but a colleague of ours, Gilles van der Wel, who's in Liège in Belgium now, he's done functional MRI to work out what parts of the brain are activated preferentially by blue light. And so he's done lots of imaging studies, both day and night time, and compared in these examples blue versus green or blue versus violet light. And he's able to show that these areas of the brain that are involved in alertness and cognition and memory and mood are preferentially activated by the blue light rather than other wavelengths. So the thalamus involved in alertness and cognition, or the brainstem again, executive function, the hippocampus for memory, and interestingly the amygdala, which is where our mood regulation occurs. Light's antidepressant effects are likely driven through light stimulus at the amygdala. So we have a photoreceptor, we know the photoreceptor system, and we know something now about the targets of light in the brain, which are exactly where you'd expect them to be if light is a stimulant, which it is. And so we've done a number of similar studies. I'm going to show you just a couple of examples of how we've looked at this system. We've compared early on a 460 nanometer blue light versus a 555 nanometer green. And the reason we chose 555 nanometers is that's the peak of your photopic 3-cone visual system. And so in these early experiments, we wanted to know, well, if your visual system is mediating this effect, green should be better than blue. If the melanopsin system is mediating this, then blue should be better than green. And so if we focus first of all on the nighttime studies, these are 6.5-hour exposures at night, a little bit like simulated shift work, if you like. So if I expose you to the same number of photons of blue versus green light, then you feel more alert at night under the blue versus green. Your reaction time is quicker, and you make fewer mistakes in your reaction time test. So blue light is a stimulus at night. It alerts the brain, and we perform better under that. Even in the daytime, we see a benefit. So we also see an increase or an improvement in reduction of lapses, and we see an improvement in reaction time in the daytime. But you'll see from in the top left, we don't see a difference in subjective alertness. And that's a little bit of a problem for us because your clients might not feel the benefit that they've just paid a lot of money to install in their space, because you're not tired enough in the daytime to necessarily feel the alerting benefits subjectively. And in fact, subjective alertness is a very poor assessment of our alertness state. But if I measure you objectively and measure your reaction time, or the brain imaging studies I just showed you, we know that blue light is alerting in the daytime as well as nighttime. And those studies, though, were with monochromatic or narrowband light. But the same principles apply to polychromatic light sources. And this is work from a colleague of ours, Christian Kaioken, and colleagues who's in Basel in Switzerland. And they've done studies looking at different polychromatic sources. And here there are three. They've got a 6500, a 3000, a 2500 Kelvin light source. And they're only exposing individuals to this light for 45 minutes, for 2 hours in the evening, ending 45 minutes before bed. And it's only about 30 lux. So this is, very conservative exposure. And consistent with the data I've already shown you, if we look at the sleepiness ratings, when you're under the bluer enriched, the higher color temperature light, you can see that sleepiness is lower. People are more alert. Their well-being, whatever that is, they feel sort of more alert and their well-being is higher. Their melatonin is suppressed, as we'd expect, more by the blue enriched white light. And their performance is improved. They are faster. So just consistent with everything I told you, if you use a higher color temperature, more blue enriched white light source, you get all those same alerting effects. But that's, of course, exactly what we don't want before bed, because this is light being given before you want to go to sleep. And so in the evening before bed, we want to look at these data the other way round, which is the one that gives you the least stimulus before bed. And so if we look at the 2500 Kelvin, the one that will have the least blue in it, you can see that then you're most sleepy under the lower color temperature. You don't feel as well, whatever that means. Your melatonin is higher. Your performance is slower. Because what we want to do before bed is take away those alerting effects of light so that you'll fall asleep quicker and have better quality sleep. And so now we start to think about, right, what are we doing in terms of the timing of light? When do we want to give a particular spectrum or a particular intensity based on the function of that space? And it's even more important than we think, because these data are looking at alertness right now, or alertness in the evening. But it has a carryover effect. So in those same experiments, they looked at sleep after the lights had been turned off. So they stopped the light 45 minutes before bed, went into dim light for 45 minutes, then went to sleep, and they measured brain activity to see what the depth of sleep was. And this is EEG, or electroencephalogram power density. What this means is slow wave sleep. So the more of this slow wave sleep you have, the more restorative the sleep is, the better the sleep is. But you can see here, after pre-exposure to the blue enriched light, you can see there was significantly less of that deep sleep, even though the lights had been turned off for 45 minutes. So just like caffeine, light has a half-life. It has an alerting effect that continues. So if you expose yourself to blue enriched light all the way to bedtime, at least for the first sleep cycle, this is about 90 minutes, probably for the second, probably for the third, although this wasn't statistically significant, it's probably just a power issue, you might be affecting your sleep quality for the next 4 or 5 hours of sleep, even though the light's off. Just like the caffeine you drink in the evening is having an impact on your sleep because of the long half-life of caffeine. So when we're thinking about how we use light, it isn't just right now, we've got to think about what light is continuing to do to the brain later on. And so it worries me when I see images like this, where people are exposing children in particular, but adults were the same, to light in the evening. Because that blue-enriched light that is emitted by electronic devices is having a major impact on our alertness and stopping our ability to fall asleep, so it takes us longer to fall asleep, we have less good quality sleep. And what's even more scary about this for children is that you only produce growth hormone during that deep sleep. So if you don't get enough deep sleep, they don't get enough growth hormone, and that may have an impact on their brain and body development. And so these are not neutral decisions when we think about the light spectrum and the light intensity and our new light sources, which are pretty new for us to think about. But it's easy to, give the kids the iPad before bed, but that's having a major impact on their biology and potentially their health. So we have to start thinking about now 2 channels. Of course, everything around design is to this point focused on that photopic function, that green channel, our ability to see. But now we've got to put everything in the context of this second blue channel as well, with this 480 nanometer peak, which is this melanopsin system. And that's an opportunity, because it means now there's something different to do. Designers are pretty good at designing spaces to help us see. We've been doing it for a long time. But now there's something new to think about and a new challenge, and I think that's really something that's exciting to think about how we light spaces. And so there are a couple of general principles that I think, how I think we should be applying light in this way. And it doesn't have to be that complicated. And a lot of people are trying to make it complicated, maybe to make money or for other reasons, but I think it's pretty straightforward. In an environment where people don't sleep, you're trying to make them as alert as possible. It's as simple as that. A school, an office, a factory, a place where they're not sleeping, you need to be alert. So for that, we should be using a high intensity, blue enriched, and by that I mean a high melanopic, a light that has a lot of that 480 nanometers in it, all the time, all day, just like the sun. The sun doesn't change in terms of its color temperature through the day. Whether you're a shift worker at night, the same thing. We want you to be safe all night. So it's all blue enriched all day. And we're using the stimulant properties of light here. We're not invoking circadian changes. We're not talking about circadian rhythms, even though a lot of people do try to. This is just alertness light as a stimulant. We don't need to tune it. For the circadian system, for the non-visual system, the sun doesn't tune. It's all blue all day, and that's what we're trying to imitate here. But some companies are saying, well, you need a blue boost in the morning, then you can let it go down, then a blue boost after lunch, then a blue boost in the evening. It's unnecessary, and it's unnecessarily expensive, unnecessarily complex. We just need to put the right lamp in the fixture so that you get that alertness all day long. And then you might see people in a workplace say, well, towards the end of the day, we want to start saying, let's bring the intensity down, let's bring the blue content down. But I don't recommend that because I don't want to do something in a workplace that makes you sleepy before you drive home. So there's no need even for that reason in a workplace or a school or a college where we would be doing that. Now, there are going to be specialized uses. If you're designing a dermatology suite in a hospital, yes, you might want to be able to change the spectrum to see different skin tones. So there may be other reasons for having a tunable system in a space. But from an alerting or a circadian point of view, it's not necessary. And so we can apply this with less expensive fixed spectrum solutions. We don't necessarily need to have an all singing, all dancing tunable system that does all these different wavelengths for a place where you're not sleeping. But where you are sleeping, then we do need the ability to change the intensity in the spectrum. That can still be simple, but we need to think about how we're going to use light. So during the daytime in a space where people sleep, it's the same. All blue enriched all day. But then when we get to the evening, and we can decide what we define as evening, but as a starting point, think of all the time after dusk, between dusk to bedtime. That would be evening, biologically. Then we want to start to dim and blue deplete or red enrich the spectrum to try and take those alerting effects away for as long as possible. And the longer the better. Your melatonin rhythm comes up about 2 hours before sleep. So if you want to make sure you don't affect your melatonin levels, then you need to start this process more than 2 hours before sleep. But I won't argue if you can tell me you can only do it for 30 minutes, because I'd rather take the 30 minutes than nothing. But the longer, the better. So then we dim it, we reduce the melanopic content, the 480, and then we're using light, or what we're trying to do there is really take away the alerting effects and help you fall asleep quicker. And as I mentioned before, stability is key. Bright days, dark nights, and as stable as we can in terms of the pattern. Now, even when we get into spaces where people are claiming circadian benefits, it isn't really the case. So if we think about care homes, I'll talk about this in a second, we're really just maintaining our light-dark cycle. We're not really shifting the clock here. And so there isn't really a circadian element to this. It's still really the alerting in the day and the sleep promoting at night. When we get into real circadian applications, then that's a specialist job. That's not going to be what most lighting designers are dealing with. Primarily, we're going to be dealing with this daytime alertness and nighttime sleep. And so we can think of this, these long blue and rich days, all blue all day, and then the dimming and the reddening in the evening. And you'll see a lot of people talk about color temperature. I'm going to show you why that's why it's the wrong unit in a moment. But again, as a shorthand, as a way to communicate, as a generalization, then we would want higher color temperatures in the daytime and then lower color temperatures between dusk and bed. And just to sort of give you a sense of some of the impact this work has have, NASA are fitting, as we speak, new lighting, new multi-LED lighting on the space station to take advantage of this knowledge. They've been sponsoring work like this for many years now. And so we have a multi-LED system where you could have a disco if you wanted. It can produce all these colors, but they just want 3 boring presettings. So we've given them those. So we've got a good visual acuity light. We've got a high circadian, high alertness boost, which I encourage you to use all the time, which is a higher color temperature, higher intensity. And then in the sleep quarters, there's a low stimulus, low color temperature, dimmer light. And so they're on the space station now. We're testing the impact of them on the space station. And so this is really space-age technology you can bring to your designs. But if we come back down to earth a little bit and think about some of the evidence for application in the real world, there are a number of examples I can show you. This is 10 years old now, and this is a study in an office setting that was done by the University of Surrey, by Dirk en Dijk and colleagues, and they did a study where they compared a 4,000 Kelvin to a 17,000 Kelvin fluorescent lamp. And I know 17,000 Kelvin sounds crazy, but it actually isn't. It doesn't look too bad. This isn't it. This is just for illustration. But over 4 weeks of each condition, and they swapped the conditions across floors in about 100 workers, they found these global benefits subjectively on alertness, performance, and sleep. And then if you look at some of the time course of these, you can see sleepiness was significantly improved, mental effort, mood, eye strain, focus, concentration, blurred vision, eye fatigue, self-reported deep sleep. All of these were shown to improve just by having the daytime exposure to the blue and rich source. And so this is a direct application of the lab work into the real world. And if we look at schools, and there's a lot of work going on in schools, the same work is being done, and here are 2 classrooms lit with different color temperature lamps. And of course, if you put them together like this, you see the differences. But if you only have that source, then it's white. It's white light. Your eye sees it as white. But the comparisons have been made in these types of settings, both with LED and with fluorescent lights. Here's a study that Osram sponsored with LED lighting, and they combined 2 different sources to come up with about a 5,500 Kelvin color temperature compared to a standard 3,000 to 4,000 Kelvin. And they did a very clever study here. They looked at the longer term effects over 5 weeks, but also looked at the effects just on 1 day, because these alerting effects are immediate and direct. And so they could see, just in 1 day, an improvement in concentration scores in a standard test in kids between the standard and the acute. And then when they measured them at the end of 5 weeks, the benefits were still there, because those effects of light are going to be there each and every day. And they're not going to die off over time. And the reason for that is that for your entire life, you need to measure day length to keep your clock synchronized. So this system doesn't die off, it will just keep going. So if you've got the light there day after day after day, you're getting the benefit day after day after day. It's not disappearing because the system's evolved to measure day length. And then Philips has sponsored a number of studies. We're not involved in these at all, but Philips have sponsored a number of different studies that are published where they've looked at fluorescent sources, but again, a very similar concept where they've had various intensities and spectra of light. But what comes out overall generally is their focus setting tends to be the one that's best, and that's 6500 Kelvin, 1000 lux. Seems crazy, but of course sunlight is much, daylight is much higher than that. And they've been able to show in multiple studies in multiple sites benefits in standard reading and concentration in these studies. And so there's certainly a case immediately for changing lighting in schools and moving away from the 3,000 to 4,000 Kelvin standard lights that tend to be used, because they're in fact stopping kids learn. And this isn't a neutral decision. This isn't about aesthetics. We need to put more of the blue enriched lights into a learning environment, because we're potentially preventing learning from happening. Now, we've also done a number of clinical studies and looked at different clinical groups where we've used blue light or blue enriched white light to treat winter depression. And if you use the right spectrum, you don't need as much of it. So traditionally, people have sat in front of giant light boxes and said you need 2,500 lux or 10,000 lux of light. You don't. If you use the right, this white light source is about 100 lux of blue enriched white light. Now, that's the wrong measure. We're going to come to that. But you don't need much if you're using the right light. And there are a number of studies that we and others have done in using light in other ways to treat fatigue. This is again using the stimulus effects of light to treat symptoms. And so, for example, patients with traumatic brain injury often have fatigue. And so one thing we've been able to do is to reduce their fatigue significantly by use of a blue enriched or blue light box in this case compared to yellow or a dim light control. Other people are doing similar studies with stroke patients or with patients receiving chemotherapy, of which fatigue is a side effect. So any space where we, or any individuals who are suffering from fatigue can potentially benefit from this. And now, so how do we take that into design? Well, your rehabilitation units could have better lighting. Your chemotherapy suite should have better lighting. Your home can have better lighting to take advantage of this. It doesn't have to be sit in front of a light box. The room can be the light box. And really good examples of where I think there's a major opportunity and we can do a lot better job is work in dementia. And this is work primarily driven by Oos van Sommeren. It's a Dutch researcher, been studying this a long time, and this is an early paper back from 1997. And they looked at the wrist activity, the sleep-wake patterns of dementia patients. And so these are from wrist monitors, and this is over 5 days. And if you look at the top panel there, it's hard to determine which is the day and which is the night. And they were in the common areas in this space, about 400 lux of standard lighting. All they did in this first experiment is just increase the intensity of the light. They didn't change the spectrum. They just increased the intensity. And you can see what happens to the sleep-wake cycle. You can see how it's much more consolidated in terms of day-night. And then they dimmed it back down again to where it was before, and the problems come back. And so just intensity increases will have a benefit. But it's going to be more efficient if you can change the intensity and the spectrum, because then you don't need as much of the light if the spectrum is correct. And so again, in 2008, this is 10 years old, and we should be applying this more. They did a clinical trial, a randomized prospective clinical trial, the gold standard you can do for a clinical study. They published it in JAMA, and they showed with the light therapy, just by increasing intensity, they got a slowing of cognitive decline as big as any drug approved for dementia, just by changing the lights. They also had less depression, they also had better sleep. So light as a drug, light as a therapy has really potentially powerful effects. And it makes sense because the brain is looking for that day-night cycle. And if you don't have it, it gets confused and the clock doesn't control rhythms appropriately and you're not as alert and you don't sleep as well. So just using light intensity, you could get this benefit. But then other researchers are also showing we can get similar benefits with increase in the blue content. This is a study from Marilyn Anderson and colleagues. They put in a 9,000 Kelvin light and increased the intensity just for the daytime in a care home until 6 o'clock. And they got clinically significant changes in sleep, in depression, and in agitation just by changing the light in the daytime. And that's not even with us changing the light in the bedrooms to help them sleep. That's just giving you that better daytime. Now, if you've got daylight, you would use daylight. I know this is a daylight group and I always forget to talk about daylight. Daylight is, of course, what we would want. And in spaces where we have daylight, let's use it because it's blue and rich and it's high intensity. But we don't have to go very far into the room before that daylight isn't really helping us in that corner. And so, yes, daylight first and foremost, but when we don't have it, we have to imitate it in a way as best we can and make sure we are getting those blue wavelengths even in the places where light doesn't get to. So just to quickly summarize that part, and then I'll just spend the last few minutes talking about some of the assessment effects. So we've got these data now. Blue light is an effective countermeasure for sleepiness at night and during the day. There are good field studies in offices, in shift work, in care homes, in schools, in space, showing that we can have these benefits in the real world by using polychromatic sources to help alertness, and then we think safety and productivity. So how do we measure and model this? There are a number of different tools that have started to think about that, thinking about how we measure this second channel. And one that I'll point out is from Lukas and colleagues. So a group of us met at the University of Manchester, where Rob Lukas is a professor. He researches these effects in animals. And this group came up with a tool that takes a spectrum, a light spectrum, and works out how much of each of the 5 photoreceptors would be stimulated by the light. Because we've got our short, and we've got our short wavelength cone, melanopsin, rods, medium and long wavelength cone. And the point of this was to try and standardize how we describe light so we can compare across studies. And that's been translated into a website by a guy called Michael Herf, who creates the software called f.lux, which some of you might have used. It's software that goes on your computer screen. It's free. And it changes your computer output in the evening, starts to dim and redden the light based on your local time. He's taken the Lukas tool, and I'm going to show you that in a second, and translated that to hundreds, if not thousands, of spectra now, so you can start to see the differences. And that's called fluxammeter.com. You can just go there. We, with Marilyn Anderson and with John and with others, have been thinking about how to translate this into the design process, and have published a few papers on that as really just proof of principle. But now we've turned this into a tool, and I'll show you in a second. We have a new software tool working with Solema, which is Christoph Reinhardt and colleagues at MIT in Boston. And now we've just released a month ago our adaptive lighting for alertness tool, which helps you design a space with this system in mind, with the circadian space in mind. And just to be clear, you'll see lots of these names, lots of descriptions of this system: circadian, non-visual, non-image forming, biological, healthy, dynamic. They're all the same. Don't let anyone tell you any of these are different. Some people are starting to say, well, this is a circadian light and this is a non-image. No, this is all the same system. We're all dealing with the same thing here. And our early work was to try and take these 2 channels and see how do we apply that? How do we apply that to a spectrum? And then how much light do we need to reach a minimum threshold of alertness? That was our initial thinking. And we've moved away from that, but just to explain, we can take any spectrum and we can put it through V lambda, or we can put it through our circadian lambda, if you like, and calculate an amount of light that stimulates that system. And then we can say, right, how much do we need to get to a threshold? And if we say we need to get 75% alertness, we need 300 lux of 555 nanometer light to get to that level. But if we've got our 17,000 Kelvin, we only need 50 lux of that light to get to the same threshold. So the idea there was that if we have a minimum threshold we want to achieve, which is always the question, how much light do I need, that designers ask, this was an attempt to think about how much we need. But I personally now think we should move away from that, because we're never going to rewrite the lighting handbook to give you thresholds or non-visual thresholds for everything we do. I think there's a simpler approach. But we've tried to take that, and we've looked at modeling by time of year, by space, by orientation, by location. We've tried to apply these non-visual responses in the same way you'd think about this for visual. But this tool that I mentioned, the Lukas tool, which is free, it's just an Excel spreadsheet, I'm happy to send it around to everyone, allows you to take a light spectrum and think how much of each of these 5 photoreceptors are you stimulating. And what it does is this: you put your spectrum in, and then it will tell you in equivalent lux, if you like, how much each of these are being stimulated. So you can see here we've got an incandescent source. Obviously that's going to be red dominated, relatively poor stimulus of melanopsin. But if we go to a 455 nanometer narrowband, you can see we're stimulating the short cone a lot, and melanopsin to some extent. What the model doesn't yet do, though, is tell you how to combine the lighting to get the best biological effect, and that's a longer story. But for the time being, what we're really interested in is how much of this melanopic lux a light source has. And so here's an example of 4 different spectra and how we might think about simplifying this process. So we can calculate the photopic lux P or the melanopic lux M, just by essentially putting your light spectrum through one of those 2 filters. And then if we take a ratio of those, M over P, we can get a sense of how alerting or not alerting that light is going to be. So if we take a standard 4000 Kelvin fluorescent and apply those 2 filters to this, we see we get a ratio of about 0.6. It's neither here nor there. It's sort of in the middle in terms of any alerting effect. If we go to a 2700 Kelvin compact fluorescent, it's around 0.4. It's a redder source, so that ratio isn't as high. But again, that's not really good enough for what we want to do. What we're trying to do, and these are initial thresholds. There's nothing hard and fast about these. This is our starting point. But a higher ratio, higher melanopic light is something with, we think, an M over P of about 0.9 or higher. Something which is going to help you sleep and have less of an impact on your alertness is something at about 0.35 or lower. And we've got some data, and other labs have got data to support some of these. But there are now, of course, commercial light sources that are designed specifically to this. So there are some commercial evening light LEDs that have a ratio of about 0.3. And that's great. We want to get that ratio down. And then if you go out and get a standard 6500 Kelvin LED, you can get a high ratio. Now, whether that's the most efficient or not, we don't know. This is peaking, at 450, 460. We want it to be peaking over here at 480 or so. So there are, again, other companies producing specialized spectra, which I approve of, that are peaking longer. It's not good enough just to take a standard blue and rich white LED and say, well, we're there, we can do a better job. But you can start to see, just through a simple ratio, we can start to get a sense of what the light is doing without changing all of the rules. You're still going to design to see how much light is hitting that surface. All the visual design is still going to happen. But what we need to add on now is just an extra layer. Say, well, what's the ratio of the light I'm using? Is it the right light source for the function I want? Without having to know anything about melanopsin or the neuroscience or any of it, you can just say, what's the ratio? Simple to calculate, is it the right source? And that's going to take us much further even than we are now. And here's a quick example of how that's being used. This is a study. It's not really a study, it's a pilot to look at care home lighting. And I don't necessarily agree with the pattern they have, but essentially they've got blue enriched days, then they dim it and make it a redder, and then in the evening it's even redder. But I'm showing this because they are calculating the melanopic ratios of these spaces. So this should be lower, but they're starting to think about how we measure this and apply it into space appropriately. And the reason CCT is not good enough, as you all know better than me, I'm sure, is that it's not specific enough in terms of the spectrum. And so here are 3 approximately 47, 4800 Kelvin color temperatures, 3 very different spectra, and 3 very different M over P ratios. So we could take the same CCT, neither here nor there, getting a bit more alerting, much better at alerting. So CCT is not good enough to specify the lighting for these functions. It's a good shorthand, and if you're trying to encourage people to buy the right things from B&Q and say you want 6500 in the day and 2700 at night, I'm happy with that for the public, but it's not good enough if we're trying to source lights to do a better job in the space. And we're just wasting energy if we're not putting it at 480. Put the energy where it needs to be, get it to that 480 peak, and then you can have a much more efficient light source and ultimately may use less energy because of that. And as I mentioned, the Flux Ammeter site is a place where you can go and play. It started as a screen, data on screens, but it's also got a lot of light sources in. And you can click on any of these and it will show you the spectrum, it will show you the melanopic lux, you can tabulate it, you can output it, you can filter it. There's all sorts of standard illuminants in there as well. But as an educational tool for yourselves and for others, it's a nice way to start to understand how different sources change in terms of their melanopic ratios. And it's interesting sometimes to find things that are not as they seem. And so very quickly, the Alpha tool, and if you want more information, you can find this on the Solema site. This is now a new lighting design software that takes all aspects of the design and thinks about it from this non-visual effect. And so it gives you at every part, and I'll show you some examples in a second, it gives you the Melanop looks and the M/P ratio of every element of the design, the paint color, the glazing, the floors, everything you need. In addition to all the other things that Alpha does or Diva for Rhino does, which is the tool. There are many other aspects of it, but Alpha is the new plugin. And the way they've done it, and this is the real advance, I think, instead of modeling lights in 3 channels, RGB, they now model light with 81 channels. So about every 5 nanometers you have data. So you're now able to model full spectrum of the light for all aspects of the design, not just our bit, all of it now, all the sky models, or everything that's involved is now an 81 channel model. So allowing to us a much more accurate modeling of all aspects of light design, not just the circadian part. So Alpha is a plug-in to Diva for Rhino that Solema produce. And here's a screenshot of how you might start to use it. So you design your space, you pick all your different parts of the design, and then it can take any part of the space in any dimension, look in any direction, and give you the spectrum that you're seeing at that point, and then give you data here. And you can't see that, but it's giving you the Melanop it looks, it's giving you the M/P ratio. It can tell you how, if you're reaching certain thresholds that you might want to put in there in your design. So it gives you lots of information here, and you can change these thresholds. If you want it to be something different you're designing to, you can change that. And it tells you, are you meeting it or not with this color coordination. So you can use our settings, but you can make your own if you want to design to a different space. And as you go looking, as you build your design, then as you choose, for example, your paint, it gives you everything there, but it's also giving you the M/P ratio. There's no point us putting a nice blue and rich light in the ceiling if we paint the walls orange, because that's not going to reflect the light we want in the eye. So the space now has to be sympathetic to the achievement we're trying to make here. So in the daytime, we want to reflect the short wavelengths or make it neutral, whereas in the evenings we may do something different. So hopefully as you're choosing and making your design, you're picking elements of the design that are sympathetic to the overall goals of the space, and then we won't end up with things that look terrible with a blue and rich light source and a red carpet. And so you can go through and you get information as you go through. You get the M/P ratio, you get the melanopic reflectance, the photopic reflectance. You can start to get a handle on how different sources, as I said, paint colors, furnishings, reflectance, all of this is incorporated. And so it's about sticking to these principles. And people don't sleep in the space, we want to encourage more of those blue wavelengths in the space. But when they do sleep, we want daytime blue and rich, but nighttime blue depleted. And just for my last slide here, in terms of thinking how we use this information, we need still a lot of education. There's a lot of misinformation out there. There's a lot of claims being made which are really not supported by data. But I think we simplify the process if we even stop talking about circadian rhythms. I'm a circadian biologist. I love circadian rhythms, but it's too complicated. And in most applications, it doesn't matter. We're not trying to shift your clock. We're just trying to alert you directly. So think of light as a stimulant, and that's a much easier message to get across to your customers. And so we can simplify the information then. And then we're not going to make claims that are not supported by saying you need this for your circadian design, it's going to go up and down, and that's what the clock wants. It's all rubbish. But it also means we don't oversell lights. It does none of us any favors if companies oversell lights that are not needed, that are too expensive, because it's not a fair application of our science, and it does us all harm because we all get tarred with the same brush. So we don't really need tunable lighting for most applications, and even in places where people sleep, we don't really need it there. And if we're going into someone's home, well, you've got a ceiling light in your bedroom and you've got a bedside table lamp. You put the blue and rich light in the ceiling for the daytime, and you have your blue depleted light in the table lamp. Your bathroom has 2 circuits, your kitchen does, your lounge does. We don't need a tunable system with an app. Just choose the right light bulb for the right place. And for most workplace settings, that's still going to be the case as well. We need to describe the light in the same way, and that's where the free Lukas toolbox is very good. We need to now get that into the design process, and that's what Alpha does. And we need to simplify this. If we make it too complicated, designers are not going to do it. customers are not going to understand it. And so taking approaches like that ratio is a simple way of trying to get a good message across without overcomplicating it. And I'll stop there and take questions. Thanks very much.
Thank you very much. I'm sure there are stacks of questions. So, please, one here.
My son's extremely light photosensitive. So he tends to try and deal with that just in terms of reducing wax, wearing sunglasses and so on. Is there anything else he should be doing?
It depends why he's light sensitive. So you know, you'd want to try and find out why that is. Certainly, there is some work that is saying that some of the light sensitivity that people have, for example, with migraine, could be through this melanopsin system. And as we learn more about this system, we're discovering that more of the non-visual effects do seem to be driven through melanopsin. So in that case, you might want to be using glasses that particularly cut out those blue wavelengths if melanopsin is, or that system is mediating these responses. But it's hard to know until you really know what's causing the issue. It's hard to have a solution. So dimming, yes, will take all wavelengths down, but there might be a way to change the spectrum of his exposure without necessarily dimming as much. And there are glasses out there that can cut blue wavelengths. Some are better than others, but you could look at the transmission and see how much of that 480 is being cut out, and that might be helpful in that area. And people are doing research to see, for example, kids who are sensitive to light, children with autism who are sensitive to light, for example. People are looking at whether that type of a solution is a good one, or changing the spectrum for very specialised sort of clinical cases like that.
Yes.
Yeah, I was wondering, you've described mostly the non-visual response due to the melanopsin receptors in the eye.
Yep.
To what extent does light falling on your skin have a different effect or a similar effect?
Yep, so that's a good question. And a number of years ago, 20 years ago now, there was a study showing that light through the back of the knee, there's a claim that light through the back of the knee could affect our clocks. That is not true. We were one of the first groups to refute that. But it wasn't as crazy an idea as we think. The heme is very similar to chlorophyll in terms of its structure, and so there was an idea that light is affecting heme in that way. And of course we treat bilirubin, or we break down bilirubin, treating jaundice by light through the skin. Vitamin D synthesis is skin driven. So certainly there are light effects, but it's not related to this system. And so we know pretty well from a number of studies that only light through the eyes affects the circadian system. And in fact, most of my career I've studied blind people, and we've shown, you know, it is the eye that you have to have, and that's true of all mammals for these systems. But there are still effects of light through the skin, and they're important. That's just not what we focus on. Yep.
There's a couple of people that I read about last year, sports psychologists or sports scientists that are using light driven through earbuds or earphones to help regulate sleep. And is that the same as the light through the back of the knee, or is that light ending up?
It's most likely a light at the back of the knee story. Again, it's not a crazy idea, because there are opsins in the brain. So there are there are molecules in the brain which might be sensitive to light. There are opsins there, but we don't really know if they are functional photoreceptors or not. So it's not an idea without any basis, but the experiments, in my opinion, haven't proved it's that. I suspect some of that light is hitting the eye in a different direction, and there's obviously placebo effects and all those other things. So no, I mean, my blind people with no eyes can't synchronize their clocks to light, and that's a pretty strong piece of evidence that it's through the eyes. Yes, sir.
You've probably heard of well standardised specs.
Yep.
I just wondered, so should employers, rather than trying to mess about with circadian light and read office environment for their day workers, would it be better simply to provide them with home guidance as to when there's suspicion on their TVs and iPads and those kinds of devices?
So I've been involved with the WELL standard. I've set some of the exam questions for those of you who have become WELL certified. And so know about that process, and I think they're taking a good approach in having people think about non-visual lighting, whether you know their numbers are correct. You know, in the first version it was 4 hours of a certain melanopic lux in the day. There's not really much basis for that, but you would meet that if you put in the right spectrum You could just put in the right light bulb and you would already meet those conditions. So I certainly think there should be a focus on a workspace or outside of the home, because we spend a lot of time there, as well as then the education around what to do at home. And we've actually put out a website through the Australian group, and I didn't show it, but I can send it again to people, which gives advice to employers and employees on simple ways to apply this in their own homes. And it really is about buying 2 different light bulbs, one for the ceiling, one for the lamps, and you use the ceiling lights in the day and you use your lamps in the evening. It can be that simple. We just need to tell people what to do, and then make sure that the products out there have that M over P ratio on, and people are making the right choices. It's amazing how people don't see lights of different spectra. We just need to tell them.
Yeah. Just on a personal level, at what point should I be switching off my TV before going?
So we all used to be worried about TVs causing insomnia, and of course you should never have a TV in the bedroom, first and foremost. No electronics in the bedroom, no laptops in the bedroom. That's rule number 1. But and we used to be worried about the TV because it was a light source. I'm not as worried now because these light sources are much closer to the eyes than the TV 10 feet away. So I'm much more concerned about light closer to the eyes. Inverse square rule, the intensity is much higher going to be there. So laptops, phones, iPads, and we know biologically that has an impact. So stop them for as long as possible before sleep, 2 to 3 hours ideally, but if you can only do half an hour, I'll take that. And then keep them all out of the bedroom and minimize that exposure in the evening. And then put on these software packages. Flux is free. Apple had Night Shift, which is not perfect, but it's better than nothing. Think about light, prioritize light, and get it dimmer and redder in the evening. Yeah.
You seem to be suggesting that as much blue light stimulus as possible is good. Is there such thing as a maybe oversaturation, like with coffee or such?
Well, it's a good question. People ask about that, and people ask about blue light hazard as well, which is something to think about. But my answer is always, well, compare it to daylight. No electric light source that meets normal safety standards produces anything like the either amount of light of any wavelength or the amount of blue than we get just by walking outside. So no, there isn't, in my opinion. The system saturates probably in the late hundreds of lux. We've done nighttime studies to show that, and similar daytimes. Because it's just night or day, on or off. The system doesn't need much more information than that. So no, I don't think so. As long as the lights are, of course, safe, and other issues are dealt with, like glare and other issues. But that's not my problem. Glare is a separate issue anyway. But it's from a circadian point of view, that doesn't matter. Yes, at the back, so and then I'll come.
It's a personal thing. I'm a retired public health engineer, water and drainage, so I don't know much about lighting. But I had a heart attack last year. I was hospitalised, and I was in a 6-bed ward, roughly about this size. And I was in a corner where, for the first 24 hours, I couldn't see daylight, and I was having panic attacks. And I asked them to move me so I could see the sky. And they did. Luckily, a bed came vacant, and they moved me, and I was okay.
Yep.
They were cheap strip lights. And I just couldn't. I could see a long way, but I couldn't see any daylight, and I was getting really panicky.
Yep. So there are actually a couple of studies showing that your risk of heart attack in a hospital is higher if you're in a poorly lit space. So your experience is consistent with some published data. And there's also a nice study where people have shown that the need for pain medication post-surgery is reduced if you're in a better daylit space. And, of course, we knew that 100 years ago. That's why hospitals were built with big windows for the light and the ventilation. And we've sort of gone inside and we've squeezed the light away. And so, yes, in a way, we're not surprised when these types of findings are made, because that light-dark cycle exposure every day is so fundamental to our biology that everything goes wrong if we mess with it. And so that absolutely clear, and that's the care home work. You give people that better stimulus, all the systems improve. And in fact, what I didn't tell you today, and it's true, all of our organs have their own clock. The heart has a clock, the liver, the lungs, the pancreas, and those clocks also become misaligned. So by having a better light-dark cycle and a better eating schedule, you can realign all your internal clocks, which matters then in terms of health. So I absolutely believe what you just said, and there's some data to show that better daylit hospital spaces improve outcome, patient outcome. Yes?
Sort of a personal question, so given that we're in the middle of London, should I be worried about too much blue light in my street lights?
It's a very good question. Street lights, yeah, this comes up all the time. You shouldn't be. We should be, of course, concerned with light pollution and wasted energy and all the other organisms that are affected by light, not just humans. And of course, we should be reducing light intensity and potentially changing the spectrum. For you personally, there's a couple of things you can do. Blackout curtains are very good. An eye mask is perfect. I've told you it's all through the eyes. So you can get rid of all the effects of environmental light by wearing an eye mask when you sleep. So it's solvable at a personal level very easily, but that doesn't mean we should be throwing all this light out into the environment. And LEDs have sort of been given a bad press around street lighting because we've taken out high pressure sodiums and put in bluer looking and bluer LEDs. But it is also the solution because you can dim those lights in a way that you really can't with the high pressure sodiums, or you can change the spectrum. And so Cambridge, Massachusetts, near where I live, they put in a 4,000 Kelvin LED and they dim it at 10 p.m. to 50% of the intensity on residential streets, and that brings that melanopic lux ratio, melanopic amount right down. And there are cities that have started to install tunable lights where you might want blue enriched good intensity until 9 p.m., 10 p.m. or on major roadways, but on residential streets late into the night you can start to dim and red, and so by 4 o'clock it's essentially an amber looking light. It looks like your high pressure sodium. So there is technology available right now to do that, and we should be thinking about that. But for you personally, blackout curtains and an eye mask will help. Yes.
Do you think that using blue light saturation for stimulus has the potential to make shift workers' problems worse? Is there a way that you can use lighting to protect these people that have no control over their life?
Yeah, so shift work's a problem. I mean, all light after dusk is unnatural. We wouldn't have evolved to see any light after dusk, and any light we do see after dusk makes the brain think it's day, and it starts that cascade of all those physiological effects. Shift work is the worst example of that because you're seeing light all night. There is no shift work without light. You know, that's what Edison gave us by making light easily available. So the problem of shift work goes beyond lighting. We should be looking in society at whether we need it. Do we really need 24-hour supermarkets and 24-hour libraries and 24-hour cafeterias in colleges? Maybe we don't. And so we should look at whether we even need to put people in that more dangerous position. But then if they have to work at night, then how do we do that? And there is a debate going on in the field about the short and long-term risks of shift work. I've talked today about using light at night as a higher intensity alertness as a stimulus because the risk of accidents at night in shift work is very high. About a 40% increased risk of having an accident or an injury at work if you work nights versus days. So each and every night you work a night shift, there's a danger to you, and we're trying to use light to help that. But there is also work showing that shift workers get more cancer in 30 years' time. Now that's been shown with the current lighting. So it's not that we're introducing a problem, it's already a problem. And so people are claiming, well, you shouldn't be using blue enriched light at night because that affects melatonin that may affect cancer risk. And that's a viable discussion, but I think that risk is lower than the each and every night risk of you having an accident in the workplace. Now, people are trying to develop lights which do both, which could alert the brain without suppressing melatonin. People are claiming that. I don't think they've got it yet. But there may be ways to be clever with the spectrum to give you the benefits of the light without necessarily some of the downside, and we're working on that.
The talk I mentioned in the States a few weeks ago, there was mention of the alerting effect of red light.
Yes.
Which kind of surprised me because I don't think there's a mechanism for that, is there?
So there is one group in the world who is claiming that red light is as effective as blue light for alerting the brain in the daytime, particularly. None of my work has supported that. I don't know of any other work outside of that one group. And as a group, you know, I'm part of many committees and consortia where we're trying to talk about lighting standards, and we're going to have that debate and come to a standard. It doesn't make sense to me for red light to be a stimulus in the day because there is no red light in the day. We've evolved in blue enriched sunlight. You don't see 4,000 Kelvin in the daytime. So it doesn't make sense to me, but there's only one group who've shown that. There's no independent validation of that work yet. But we'll keep an open mind. So I've spoken to you, but I just want, I know we have to stop. We have another speaker, but at the back, lady.
In the olden days,
Yes.
did people, I think some people used to get up in the middle of the night. So are there certain chronotypes that like a bit of awakeness during the night?
Yep, so what you're referring to is the idea that we had a much more natural light dark cycle. We'd go to bed earlier because the lights went down and we couldn't light the night. And then maybe wake up in the middle, then go to sleep. And Pepys talked about that in Pepys' diaries. He talks about going out and walking the streets at night. And maybe that was the case. But in practice, it doesn't really work for us anymore because any electric light after dusk is pushing our clocks later. And so we end up squeezing the night into a shorter time because we don't shift dawn later, but we shift our own dusk later because we use electric light in the evening. And a colleague of mine, a guy called Ken Wright, who is at University of Colorado, he's taken people camping in the Rockies without any electric light source and shown that within a week he can completely correct that imbalance if you only have natural light. And what happens then is you fall asleep earlier, you sleep for longer, and that melatonin rhythm adjusts earlier so that the melatonin peak occurs at midnight. Because midnight used to be the middle of the night. And it's not anymore. So we can do it, we still have the biological capacity, but you'd really have to take away any electric light sources after dusk for about a week. You'd adjust yourself to that, and then you probably would fall asleep earlier, you'd probably wake up, get up, you'd probably go back to sleep. But are you really going to do that for 14 hours in the winter? Probably not, and so we could do it, but I don't think it's going to be that helpful to us.
I think we've got time for one more question.
Sorry.
Actually, the gentleman in the poorly lit corner of there.
Yeah, fast asleep.
So continuing with the question which she asked, in India, for example, there are offices which run 24/7, and you have people who are exposed to displays and computers during nighttime. So having high intensity ceiling lights along with the blue lights emitted from the displays, is it beneficial because it will actually impact them?
So that might be a different application. So yes, if you've got a great big screen in front of you, that's your light source. That's what it's hitting the eyes. And so maybe then you don't need to supplement. But when we look at visual function, actually it's better for us to have a bright background with a bright source. What you don't want is that huge contrast. So in fact, having brighter screens means we can have brighter backgrounds as well. And so it's not just that you get it when you're sitting there, but when you walk to the cafeteria and all the spaces you're in, you can still be getting that same benefit. And you can even turn that round. So we did one study with NASA Mission Control. You see, on TV you see that Mission Control picture, all those big screens. They're not looking at any of those big screens, that's just for show. They're looking at the screens on their desks. But they wouldn't change the lighting in there because they didn't want to change the imagery of the space. But we filled the break rooms and the corridors with light. And so when they'd get their break every 2 hours, they'd get a pulse in the break room or the corridors, which gave them enough of a boost for the next couple of hours. I've shown you those alerting effects continue. And so, you can think about if you can't do it in the space you want and have to do it somewhere else, you can have that sort of light shower every so often, or allow people to have brighter screens to see better, but with a brighter background, so you reduce the contrast. But we should end there. But thank you very much indeed. Thanks.